Good evening and welcome to Dreamy Academy. As we begin, I'd love to know where you're watching from. Your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Right now, as you listen, you are not alone, not truly. Within you, upon you, and all around you, an invisible universe teems with life, a world so ancient, so diverse, and so profoundly influential that our own existence is woven into its very fabric. We often perceive the world through the scale of our own bodies, the sweep of a landscape, the reach of a hand, the faces of those we love. But beneath this familiar surface lies a realm of exquisite complexity, of tiny architects and silent engineers, living out their intricate lives in numbers that defy comprehension. To journey into this microscopic world is to understand not just the smallest components of life, but the foundational principles that have shaped every breath, every ecosystem, every evolutionary leap across billions of years. It is a journey into the very essence of what it means to be alive, a quiet contemplation of the ceaseless dance of matter and energy that unfolds in every drop of water, every grain of soil, and indeed within every cell of your own being. This is the story of microbiology, a story of wonder that lulls through beauty and depth, inviting us to see the profound in the infinitesimal and to find peace in the intricate tapestry of life. Picture, if you will, a world without eyes, a world where senses are chemical, where movement is a silent glide, and where the very air is a vast ocean of possibilities. This is the world of the microbe, a realm that existed for billions of years before the first visible creature stirred on Earth. Tonight, we begin our quiet journey into this hidden foundation of life, understanding how the smallest entities orchestrate the grandest patterns. Microbes, or microorganisms, are simply living things too small to be seen with the naked eye. They are the invisible majority, the silent architects of our planet. When we speak of them, we are speaking of an astonishing diversity. Bacteria, archaea, fungi, protists, and even viruses, though viruses exist at the very edge of what we define as life. To grasp their scale, imagine taking a single strand of your hair. A bacterium, on average, would be about 100 times smaller than the width of that hair. A virus, smaller still, could be 1,000 times tinier than a bacterium. These are worlds within worlds, operating at a level far beneath our everyday perception. Yet, despite their minuteness, their presence is pervasive. They thrive in the scorching vents of the deep sea where no sunlight penetrates, and in the frozen desolation of the polar ice caps. They float in the clouds, drift in the deepest ocean trenches, and permeate every millimetre of soil beneath our feet. A single teaspoon of rich garden soil can contain more microorganisms than there are people on Earth. Your own skin, your mouth, your gut, each is a bustling metropolis for trillions of these tiny beings, a vibrant ecosystem where countless interactions unfold every second. They are not merely around us, they are an intrinsic, inseparable part of us, and of the living planet itself. Their story is the oldest story of life, echoing through the eons, To understand these unseen architects, we must first appreciate a fundamental distinction in the architecture of life itself: the difference between prokaryotes and eukaryotes. It is a distinction that began billions of years ago and continues to shape every living thing on Earth. The simplest and oldest form of cellular life is the prokaryote, a term that, in its quiet Greek roots, means before nucleus. Imagine a tiny, self-contained bag of living chemistry, enclosed by a delicate membrane. Within this membrane, all the essential machinery of life, the genetic material, the protein-making factories, the energy converters, floats freely in the cell's interior. There is no distinct membrane-bound compartment to house the precious DNA. This elegant simplicity is not a flaw, but a profound strength. It allows for incredible efficiency, rapid reproduction, and adaptability. Our own cells, and the cells of plants, animals, and fungi, are eukaryotes, true nucleus. They are grander, more complex structures, with their genetic blueprint neatly packaged within a central command center, the nucleus. Beyond that, eukaryotic cells are filled with a constellation of specialized membrane-bound organelles, tiny organs like mitochondria, which generate energy, or chloroplasts, which capture sunlight. These internal divisions create a sophisticated internal architecture, allowing for greater specialization and the emergence of multicellular life. But it is the prokaryotes, with their ancient, elegant simplicity, that laid the groundwork for everything that followed. They are the fundamental blueprint, the original design from which all other complexity has slowly unfolded. This distinction, the presence or absence of a nucleus, is not merely an anatomical detail. It represents two profoundly different strategies for life, two ancient lineages that have shaped the planet in their own ways. Prokaryotes, with their directness and speed, dominate the sheer numbers of life forms and their collective metabolic power. Eukaryotes, with their internal complexity, enabled the evolution of larger multicellular organisms, leading to the vast diversity of plants and animals we see today. Yet, even in our own complex bodies, the echoes of this ancient divide persist, and the prokaryotic world continues its quiet, essential work. Having glimpsed the foundational architectures, we might quietly ask, how do these tiny beings truly live? How do they sense, move, and thrive without the complex organs we recognize? That question will lead us into the elegant mechanisms of their cellular lives. As we gently concluded our last exploration, a profound question lingered in the quiet corners of our contemplation. How do these tiny, unseen architects of life, these microbes, truly live, sense and move without the complex organs and systems we associate with larger forms of life? It is a question that invites us to peer even deeper, beyond their mere presence, into the very heart of their being. We are about to embark on an intimate journey into the cellular world, to understand the elegant simplicity and profound strength that define these foundational entities. Imagine, if you will, a miniature self-contained universe, bustling with purpose, exquisitely designed for survival and propagation. This is the prokaryotic cell, the most ancient and prevalent form of life on our planet, a testament to efficiency and adaptive brilliance. We speak of bacteria and archaea here, those pioneers who laid the very groundwork for all subsequent biological complexity. Their secret, as we shall soon discover, lies not in grand structures, but in the ingenious orchestration of fundamental components each playing a vital role in their enduring success. Let us begin our journey at the very boundary of this microscopic world, the cell wall. For many prokaryotes, this is their first line of defense, a sturdy, often rigid outer layer that provides structural support and protection against environmental stresses. Think of it as the resilient outer shell of a tiny living fort, maintaining the cell's shape and preventing it from bursting in environments where water might rush in. It is a protective embrace, custom engineered for the specific world the microbe inhabits, a testament to billions of years of evolutionary refinement. Just beneath this protective wall lies the cell membrane, a marvel of selective permeability. This delicate yet incredibly dynamic phospholipid bilayer is the true gatekeeper of the cell, a shimmering boundary that carefully regulates the passage of substances in and out. It is here that the cell truly interacts with its environment, sensing changes, importing nutrients, and expelling waste. But its role extends far beyond mere gatekeeping. For many prokaryotes, the cell membrane is also the primary site for critical metabolic processes, including the generation of energy. It is a biological powerhouse, a miniature factory floor where the vital work of life unfolds. Step inside this membrane and you enter the cytoplasm, the vibrant gel-like substance that fills the cell. This is the inner ocean, the bustling metropolis where countless biochemical reactions occur simultaneously. Within this aqueous environment, you'll find essential ions, nutrients, and a myriad of enzymes, all working in exquisite synchronicity. It is the stage upon which life's intricate play is continuously performed, a testament to the dynamic nature of even the simplest cells. Scattered throughout this cytoplasmic expanse are tiny, granular structures known as ribosomes. These are the cell's protein synthesis factories, the tireless workers that translate genetic instructions into the functional proteins essential for every cellular activity. Without these miniature machines, the cell would be unable to construct the enzymes, structural components, and regulatory molecules it needs to live, grow, and reproduce. They are universal, found in all forms of life, a profound echo of our shared ancestry. And then, at the very heart of this prokaryotic universe, we find the nucleoid region. Unlike the neatly packaged DNA within a eukaryotic nucleus, the prokaryotic genetic material, typically a single circular chromosome, is not enclosed within a membrane. Instead, it forms a dense, irregularly shaped mass within the cytoplasm. This is the cell's master blueprint, the repository of all the information required for its existence. It is here that the intricate code of life is stored, replicated and expressed, guiding every function, every adaptation. But the story of prokaryotic genetics often includes another fascinating element: plasmids. These are small, circular pieces of extrachromosomal DNA that exist independently of the main chromosome. Think of them as supplemental instruction manuals, often carrying genes that confer advantageous traits, such as antibiotic resistance or the ability to metabolize unusual nutrients. Plasmids are remarkably mobile. They can be readily exchanged between bacteria, even across different species, a powerful mechanism for rapid adaptation and evolution in ever-changing environments. This ability to share genetic information so fluidly is a key reason for the incredible resilience and adaptability of the microbial world. Now, Let us consider how these self-sufficient entities interact with their surroundings, how they move through their micro-universes. Many prokaryotes possess remarkable appendages that enable motility and adhesion. The most iconic of these are the flagella, singular flagellum. Imagine a tiny rotating propeller emerging from the cell surface, driven by a molecular motor of astonishing complexity. These whip-like structures allow bacteria to swim with purpose, navigating their aqueous worlds in search of nutrients or away from harmful substances. It is a testament to natural engineering, a biological engine of incredible efficiency. Other common appendages are pili, singular pilus, and fimbriae. These are shorter, hair-like structures that extend from the cell surface, while fimbriae are primarily involved in adhesion, allowing microbes to stick to surfaces, whether a rock, a leaf, or the lining of your gut. Pili have a more specialized role. Some pili, known as sex pili, facilitate a process called conjugation, a form of genetic exchange where a plasmid can be transferred directly from one bacterium to another. This direct sharing of genetic information further underscores the dynamic and interconnected nature of microbial populations. Having explored the intricate architecture of the prokaryotic cell, we naturally turn to the question of sustenance. How do these miniature architects power their bustling internal worlds? The answer lies in a breathtaking diversity of metabolic strategies, far surpassing the energy acquisition methods of macroscopic life. Prokaryotes are the true alchemists of our planet, capable of harnessing energy from an astonishing array of sources. Some are autotrophs, meaning they produce their own food. Among these we find the photoautotrophs like cyanobacteria, which perform photosynthesis. They capture the sun's energy, transforming it into chemical energy, much like plants do. But they were doing so billions of years before the first plant ever emerged, fundamentally altering Earth's atmosphere by releasing oxygen. Then there are the chemoautotrophs, a truly remarkable group that derives energy not from light, but from the oxidation of inorganic chemical compounds such as hydrogen sulfide, ammonia, or iron. These microbes thrive in extreme environments, deep-sea hydrothermal vents, acidic hot springs, or even within rocks, forming the base of entire ecosystems where sunlight never penetrates. They are the silent engineers of the deep, converting raw chemicals into the very fabric of life. The vast majority of prokaryotes, however, are heterotrophs, obtaining their energy by consuming organic compounds produced by other organisms. They are the great recyclers of our planet, breaking down dead organic matter, returning essential nutrients to the soil and water, and driving the biogeochemical cycles that sustain all life. From the bacteria in your gut that help digest your food to the decomposers that ensure the constant renewal of ecosystems, these heterotrophic microbes are indispensable. Their metabolic versatility is staggering, allowing them to break down nearly any organic compound imaginable, a testament to their adaptive genius. And how do these incredibly efficient cells multiply? Through a process of elegant simplicity called binary fission. Unlike the complex cell division, mitosis, seen in eukaryotes, binary fission is a straightforward process where a single prokaryotic cell grows, replicates its circular DNA, and then divides into two identical daughter cells. This process is remarkably rapid, with some bacteria capable of dividing every 20 minutes under ideal conditions. Imagine the exponential growth. This rapid reproduction, combined with their ability to exchange genetic material, is a powerful engine for evolution, allowing microbial populations to adapt swiftly to changing environments and exploit new ecological niches. It is the secret to their enduring dominance and omnipresence. Finally, we return to the question of sensing and movement. How do these cells, without eyes or ears or brains, perceive their world? They do so through a sophisticated array of receptors embedded in their cell membranes and cytoplasm, which detect specific chemical signals or physical stimuli. This allows them to engage in processes like chemotaxis, moving toward beneficial chemicals like nutrients, or away from harmful ones like toxins. Similarly, phototaxis enables photosynthetic microbes to orient themselves toward optimal light conditions. These responses are not random. They are guided, purposeful movements driven by the intricate molecular machinery of the flagella and the subtle shifts in their internal chemistry. They are constantly listening to their environment, making micro-decisions that ensure their survival. As we conclude this intimate look at the prokaryotic cell, we begin to grasp the profound answer to our initial question. They live, sense, and move, not through complex organs, but through a masterful orchestration of fundamental cellular components, each performing its task with unparalleled efficiency. Their simplicity is a deceptive term, for within their compact design lies a complexity of function that has allowed them to thrive for billions of years, adapting to every conceivable corner of our planet. This elegant simplicity stands in stark contrast to the eukaryotic cell, which we, and indeed all multicellular life, embody. Recall that eukaryotes possess a true nucleus, a membrane-bound organelle housing their genetic material, and a suite of other specialized membrane-bound organelles like mitochondria, the powerhouses, chloroplasts for photosynthesis in plants and algae, endoplasmic reticulum, and Golgi apparatus. This sophisticated internal architecture allows for greater specialization of function within the cell, enabling the emergence of larger, more complex organisms and the intricate dance of multicellular life. But it is crucial to remember that this eukaryotic grandeur arose from, and continues to depend upon, the foundational blueprints laid down by their prokaryotic ancestors. Thus, the prokaryotic cell is not merely a primitive predecessor. It is a masterpiece of biological engineering, a self-sufficient world unto itself. Its efficiency, adaptability, and metabolic versatility are the engines that drive Earth's essential cycles, shaping the very air we breathe, the soil beneath our feet, and the intricate ecosystems that sustain us. They are the quiet, persistent pulse of life, the fundamental units whose tireless work allows our own complex existence to unfold. As we move forward, we will continue to uncover the extraordinary roles these tiny beings play, reminding us always of the deep, unseen interconnectedness that binds all life on this wondrous planet. We have journeyed through the intricate architecture of the prokaryotic cell, marveling at its elegant simplicity and the profound strength hidden within its components. We've seen how these tiny architects move with purpose, sense their surroundings, and metabolize the very essence of their world, orchestrating life's fundamental processes through rapid binary fission. Yet to truly grasp the omnipresent influence of these microscopic beings, we must now turn our gaze outward to the boundless canvas upon which they paint their existence. the diverse and often extreme environments of our planet. Imagine for a moment, if you will, the sheer audacity of life. Think of the places you might consider utterly inhospitable, utterly barren. Perhaps the searing heart of a volcanic vent, or the crushing darkness of the deepest ocean trench. Maybe the crystalline silence of polar ice, or the acrid sting of an acid lake. For us, these are realms of extremity, zones where survival seems impossible. But for our microbial companions, these are not just habitats. They are homes, meticulously engineered niches where life in its most ancient forms continues to flourish, often in astonishing abundance. This chapter, dear explorer, invites us to contemplate the extraordinary resilience and adaptability of microbes, to understand how they have not merely survived, but have mastered every conceivable corner of Earth. We will discover the extremophiles, life forms that thrive where others perish, pushing the very boundaries of what we once believed possible for biological existence. From the familiar soil beneath your feet, teeming with a vibrant, unseen metropolis, to the very air you breathe, carrying microscopic travellers on unseen currents, microbes are everywhere. They are the silent partners in the roots of every plant, the diligent recyclers in every drop of water, the unseen guardians and occasional provocateurs within your own body. But it is in the realms of true extremity that their ingenuity truly shines, revealing the deep ancient wisdom encoded within their fundamental blueprint. Let us begin our exploration with temperature, a parameter that dictates so much of life as we know it. For most complex organisms, a narrow band of warmth is essential. Too cold, and molecular processes slow to a halt, water freezes, and cells rupture. Too hot, and proteins denature, DNA unravels, and membranes dissolve. But not for our microscopic pioneers. Consider the thermophiles and hyperthermophiles, the lovers of heat, who find their paradise in conditions that would instantly sterilize our world. We speak of the scalding waters of Yellowstone's hot springs, where vibrant microbial mats paint the landscape in impossible hues of yellow, orange, and green, each color a testament to different species thriving at different temperatures. Here, microorganisms perform photosynthesis or chemosynthesis in water, approaching or even exceeding the boiling point. Deeper still, at the black smokers of the mid-ocean ridges, where superheated water, rich in dissolved minerals, erupts from the Earth's crust, hyperthermophiles thrive at temperatures well over 100 degrees Celsius, 212 degrees Fahrenheit. Their enzymes, marvels of molecular engineering, remain stable and active, even in these infernal conditions, allowing them to catalyze life's reactions with unwavering efficiency. These are not mere survivors, they are thrivers, their metabolism optimized for a world of perpetual warmth. At the opposite end of the spectrum we find the psychrophiles, the lovers of cold. Picture the vast frozen expanse of the Arctic and Antarctic, the crushing depths of the deep ocean where temperatures hover just above freezing. Here, psychrophiles abound, their specialized cell membranes remaining fluid and their enzymes active in conditions that would turn most biological systems into inert ice. They are the unseen engines of polar ecosystems, breaking down organic matter, cycling nutrients, and proving that life can persist, even flourish, in the stark beauty of perpetual frost. Next, let us delve into the realm of pH, the measure of acidity or alkalinity. Most life prefers a neutral pH, a delicate balance. Deviate too far, and the intricate dance of proteins and nucleic acids can be disrupted, leading to cellular collapse. Yet across our planet there exist environments that defy this preference. The acidophiles, for instance, call environments like acid mine drainage or volcanic lakes their home. Imagine a lake with a pH close to that of battery acid, a place where metal pipes corrode within days. In these seemingly caustic landscapes, acidophilic bacteria and archaea not only survive but actively metabolize, often contributing to the very acidity of their surroundings. Their cell membranes and internal cellular machinery are uniquely adapted to maintain a near-neutral internal pH, even as the external environment rages with acidity, a testament to sophisticated proton pumps and specialized lipid compositions. Conversely, the alkaliphiles revel in highly alkaline environments such as soda lakes, where the pH can soar to 10 or 11, similar to household bleach. These microbes have developed ingenious ways to cope with the scarcity of protons and the abundance of hydroxide ions, maintaining their internal balance against overwhelming external forces. Then there is salinity, the concentration of salt. For most cells, high salt concentrations are lethal, drawing water out through osmosis and dehydrating the cell. But for the halophiles, the lovers of salt, a high salt environment is a prerequisite for life. Think of the Dead Sea, so saline that nothing larger can survive, or the vibrant pink and red salt flats around the world. These vibrant hues are often the signature of halophilic microbes, particularly archaea, which thrive in waters saturated with salt. They employ various strategies such as accumulating compatible solutes within their cells to balance osmotic pressure, or having specialized proteins that function optimally in high salt conditions. Their very existence challenges our intuitive understanding of life's requirements. Beyond these more common extremes, microbes also conquer realms of immense pressure, radiation, and even oxygen deprivation. Barophiles, or piezophiles, inhabit the abyssal plains and trenches of the deep ocean, enduring pressures thousands of times greater than at sea level. Their proteins and membranes are specifically structured to withstand these crushing forces, functioning perfectly where our own cells would be instantly obliterated. And what of radiation, that invisible force capable of tearing apart the very fabric of DNA? The radio-resistant microbes such as Deinococcus radiodurans possess extraordinary DNA repair mechanisms, allowing them to stitch their genomes back together even after receiving doses of radiation that would be lethal to almost any other organism. They offer tantalizing clues not only about survival in harsh environments, but also about the potential for life beyond Earth, in places exposed to cosmic radiation. Finally, let us briefly return to the breath of life itself. Oxygen. While many organisms, including ourselves, are utterly dependent on oxygen for survival, we are aerobes, many microbes find it toxic. The anaerobes thrive in environments devoid of oxygen, utilizing alternative electron acceptors for their metabolic processes. Some are obligate anaerobes, meaning oxygen is a deadly poison, while others are facultative anaerobes, capable of switching their metabolism depending on oxygen availability. This fundamental distinction highlights the incredible metabolic versatility of the microbial world, and reminds us that life on early Earth was almost entirely anaerobic before oxygen became abundant. What allows these microscopic beings to achieve such feats of survival? It is a combination of the fundamental cellular components we explored in Chapter 2, refined and specialized over billions of years of evolution. Their cell walls and membranes are often modified to withstand extreme temperatures or pH. Their enzymes, those molecular workhorses, are exquisitely adapted to function under specific, often harsh, conditions. They possess unique metabolic pathways, protective pigments, and sophisticated repair mechanisms that allow them to thrive where other life forms would simply cease to be. These extremophiles are not merely biological curiosities. They are living relics, windows into Earth's ancient past. Many of these organisms represent some of the oldest lineages on our planet, thriving in conditions that might well have mirrored the early Earth, before the atmosphere was oxygen-rich, before complex multicellularity emerged. By studying them, we gain profound insights into the origins of life itself and the incredible evolutionary pressures that shaped our world. Their pervasive presence in these extreme environments underscores a crucial truth. Microbes are the ultimate pioneers, the fundamental bedrock upon which all other life depends. Their capacity to inhabit every conceivable niche ensures that biogeochemical cycles, the continuous movement of elements like carbon, nitrogen and sulfur through the Earth's systems, are maintained even in the harshest conditions. They are the silent engineers, tirelessly recycling nutrients, breaking down pollutants, and transforming the very chemistry of our planet. As we conclude this chapter, reflect on the vastness of the microbial realm. It is not confined to the visible, comfortable world we perceive. It is an invisible universe that stretches from the frozen poles to the volcanic depths, from acidic springs to hypersaline lakes, a testament to life's indomitable will and astonishing adaptability. The elegant simplicity of the prokaryotic cell, far from being a limitation, is its greatest strength, allowing for a flexibility and resilience that has enabled it to colonize, transform, and sustain our planet for billions of years. In understanding these microbial masters of adaptation, we deepen our reverence for the intricate tapestry of life and the foundational role of its smallest, most ancient threads. Next, we will venture even deeper into the intricate web of life, exploring how these individual microbial pioneers interact with each other and with larger organisms, shaping ecosystems and influencing the very health of our world. The journey, dear student, continues. Welcome back, dear explorer, to Dreamy Academy. In our previous journey, we marveled at the astounding resilience of individual microbes, those masters of adaptation that defy the harshest conditions, reminding us of life's tenacious spirit. We saw how a single, elegant cell with its core machinery could thrive where others falter, shaping Earth's very chemistry from the deepest vents to the highest ice caps. But life, as we know it, rarely exists in isolation. Even the most solitary extremophile is part of a larger unseen tapestry. Today, we invite you to shift your gaze from the singular cell to the grand symphony it performs within its community. We will explore the intricate dance of microbial ecosystems, where countless tiny architects work in concert, not only shaping the planet's fundamental cycles, but also weaving themselves into the very fabric of every living being, including you. Imagine, if you will, that the invisible universe we've been exploring is not merely a collection of isolated stars, but a bustling metropolis, a vibrant coral reef, or a dense, ancient forest. Each microbe, no matter how small, is a participant, an actor in a drama of profound ecological significance. They form communities, these silent engineers, often adhering to surfaces in complex multi-layered structures we call biofilms. Think of the slippery film on a river stone, the plaque on your teeth, or the intricate layers of microbial life thriving in a hot spring. These are all testament to the collective power and collaborative nature of microbes. Within these biofilms, cells communicate, share resources, and even protect one another, exhibiting a form of collective intelligence that transcends their individual simplicity. They create their own microenvironments, transforming the world around them through their combined metabolic prowess. It is through these vast interconnected communities that microbes orchestrate Earth's most fundamental processes, the grand biogeochemical cycles that have sustained life for billions of years. These are the planetary breathing rhythms, the silent continuous transformations of elements, essential for all living things. Without our microbial partners, these cycles would grind to a halt, and life as we know it would cease to exist. Let us first consider the carbon cycle, the very backbone of organic life. Carbon, the building block of all biomolecules, journeys through the atmosphere, oceans, soil, and living organisms. Who are its primary drivers? Our microbial friends. Cyanobacteria and other photosynthetic microbes in the oceans perform vast amounts of photosynthesis, capturing atmospheric carbon dioxide and converting it into organic matter, just as plants do on land. They are the initial fixers, pulling carbon from the air and water, making it available to the food web. When plants and animals die, or when any organic matter needs to be recycled, bacteria and fungi step in as nature's ultimate decomposers. They break down complex organic compounds, releasing carbon dioxide back into the atmosphere through respiration, or locking it away in the soil. Other specialized microbes like methanogens, a type of archaea, produce methane, CH4, in oxygen-deprived environments, a potent greenhouse gas, while methanotrophs consume it, maintaining a delicate balance. This continuous, microbial-driven dance of carbon sequestration and release is a critical regulator of Earth's climate and the very availability of life's essential fuel. Then there is the nitrogen cycle. Perhaps the most intricate and vital of all. Nitrogen gas, N2, makes up nearly 80% of our atmosphere, yet in this gaseous form, it is largely unusable by most organisms. It must be fixed, converted into more reactive compounds like ammonia, nitrates, or nitrites, before it can be incorporated into proteins, nucleic acids, and other essential molecules. This monumental task falls almost exclusively to microbes. Nitrogen-fixing bacteria, such as those in the genus Rhizobium that live in symbiotic association with legume roots, or free-living bacteria like Azotobacter, possess the unique enzymes to convert atmospheric N2 into ammonia, NH3. This is a truly energy-intensive process, a molecular feat that underpins the productivity of entire ecosystems. From ammonia, other microbial groups take over, nitrifying bacteria. Oxidize ammonia into nitrites, NO2-, and then nitrates, NO3-, forms readily assimilated by plants. Finally, denitrifying bacteria complete the cycle, converting nitrates back into nitrogen gas, returning it to the atmosphere. This continuous, multi-step microbial process ensures that life's essential nitrogen is constantly recycled and made available, a testament to the profound, unseen work happening beneath our feet and within the soil. The Ga-sulfur cycle also relies heavily on microbial transformations. Sulfur, a component of proteins and vitamins, moves through various oxidation states, from hydrogen sulfide, H2S, to elemental sulfur, S, to sulfates, SO42-. Different groups of bacteria and archaea facilitate these conversions. Sulfur-oxidizing bacteria, often found in hydrothermal vents or anoxic sediments, convert toxic hydrogen sulfide into sulfates, while sulfate-reducing bacteria do the reverse. This microbial ballet maintains the availability of sulfur for biological processes and helps detoxify certain environments. And let us not forget the iptesis cycle. Though it is primarily driven by geological processes, microbes play an important role in making phosphorus available to life. Phosphate, critical for DNA, RNA, and ATP, is often locked away in insoluble minerals. Phosphate-solubilizing microbes secrete organic acids that can release phosphate from these minerals, making it accessible for plant uptake. They are the silent miners, ensuring this vital nutrient continues its journey through the web of life. These cycles, dear student, are not abstract concepts confined to textbooks. They are happening right now, a continuous, invisible labour performed by billions upon billions of microbes, sustaining the very breath you take, the food you eat, and the ground you walk upon. They are the Earth's primary alchemists, tirelessly transforming elements, shaping the planet's surface and atmosphere over geological timescales. Beyond these grand, planetary-scale transformations, microbes also engage in more intimate interactions with other organisms, forming a spectrum of symbiotic relationships. Quote 5, meaning quote 6, describes the close and often long-term associations between different species. These interactions can range from mutually beneficial to outright harmful, and microbes are masters of all forms. Consider mutualism, where both partners benefit. A striking example lies beneath the waves, in the radiant coral reefs. The vibrant colours of coral come from their symbiotic partners, microscopic algae called zooxanthellae, living within the coral polyps. The algae photosynthesize, providing the coral with energy and nutrients, while the coral provides a protected environment and compounds for photosynthesis. On land, the nitrogen-fixing rhizobium bacteria in legume roots are another classic example. The bacteria receive sugars from the plant, and in return provide fixed nitrogen essential for plant growth. Even lichens, those hardy organisms clinging to rocks and trees, are a mutualistic partnership between a fungus and an alga or cyanobacterium, each providing what the other lacks to survive in harsh environments. Then there is commensalism, where one organism benefits, and the other is neither helped nor harmed. Many of the microbes living on your skin, for instance, are commensals. They feed on dead skin cells and oils, finding a comfortable home without generally affecting your health. Finally, we encounter parasitism, where one organism, the parasite, benefits at the expense of the other, the host. This is the realm of pathogens, the disease-causing microbes we often hear about. Viruses, bacteria, fungi, and protists can all act as parasites, causing illnesses ranging from the common cold to devastating diseases. Yet, even here, the line can be blurry. A microbe that is parasitic in one context might be commensal or even mutualistic in another, highlighting the dynamic and context-dependent nature of these relationships. These symbiotic relationships, particularly mutualism and commensalism, lead us to perhaps the most intimate and profound microbial ecosystem of all, the human microbiome. Step with us now and consider your own body not just as a singular entity, but as a vast, teeming planet, a personal microbial universe. From the moment you were born, you began to accumulate a complex community of microorganisms that now outnumber your own human cells by a factor of 10 to 1. Your skin, your mouth, your lungs, and most profoundly your gut, are home to trillions of bacteria, archaea, fungi, and viruses living in an intricate balance. This inner wilderness, your microbiome, is not merely a collection of passive passengers. It is an active, indispensable organ, profoundly influencing your health and well-being. In your gut, for example, your resident microbes are busy breaking down complex carbohydrates that your own enzymes cannot digest, extracting vital nutrients and synthesizing essential vitamins like K and several B vitamins. They produce short-chain fatty acids that nourish your gut lining and influence your metabolism. They train and modulate your immune system, teaching it to distinguish between friend and foe, preventing overreactions to harmless substances, and mounting effective defenses against true pathogens. They form a protective barrier, occupying niches and competing for resources, thereby preventing harmful microbes from colonizing and causing disease, a phenomenon known as competitive exclusion. Emerging research even suggests that our gut microbes can communicate with our brain, influencing mood, behavior, and even cognitive function through complex biochemical pathways. They are silent partners in virtually every aspect of our physiology, from energy harvest to immune defense, from nutrient synthesis to mental clarity. So you see, dear friend, you are never truly alone. You are an ecosystem, a walking, talking testament to the power of microbial symbiosis. Your very existence, your health, your vibrancy is inextricably linked to the billions of tiny lives within and upon you. They are the unseen architects of your inner world, just as they are the silent engineers of the planet. As we conclude this chapter, let us reflect on the profound interconnectedness that microbes reveal. From orchestrating global cycles that sustain all life to forming intimate, indispensable partnerships within our own bodies, these tiny beings demonstrate that cooperation, adaptation, and community are fundamental principles of existence. We are not separate from the microbial world. We are an integral part of it, a vibrant testament to its enduring power and its quiet, ceaseless work. In the quiet hum of this realization, we find not only scientific fact but a deep, humbling wonder. The journey continues, and next we will explore what happens when this delicate balance is disturbed, and how we, in turn, interact with this ubiquitous microbial world. Until then, carry this newfound appreciation for your unseen partners and for the magnificent, intricate dance of life they perform. In our last shared reflection, we pondered the exquisite balance of the human microbiome, that indispensable ecosystem within and upon us, and asked a profound question. What happens when this delicate balance is disturbed? Indeed, dear explorer, this query leads us into a realm where the unseen architects, usually our allies, can sometimes become formidable adversaries, challenging the very foundations of our well-being. For all their ancient wisdom and benevolent roles, not all microbes exist in perfect harmony with larger life forms. Some, though a minority, possess the capacity to disrupt, to cause illness, to become what we term pathogens. These are the microbes whose strategies for survival and propagation, when enacted within a host, can lead to disease. Yet it is crucial to remember that this is not a simple tale of, quote, 7 versus, quote, 8. Often the line is blurred, for even our most cherished microbial residents can, under altered circumstances, a weakened immune system, a shift in location, an overgrowth, transform into opportunistic pathogens, seizing a moment of vulnerability to flourish at our expense. It is a testament to the dynamic, ever-negotiating nature of life itself. How then do these microscopic entities exert such influence? The journey from harmless presence to disease-causing agent is a complex ballet, orchestrated by what we call virulence factors. These are the specific traits, the cellular tools and biochemical pathways that enable a pathogen to establish itself, overcome host defenses and cause damage. Imagine, if you will, a microscopic invasion. The first critical step for any potential pathogen is adhesion, the ability to firmly attach to host tissues. Without this anchor, they would simply be swept away by the body's natural currents, expelled by the flow of fluids, the beating of cilia, or the constant shedding of cells. Bacteria, for instance, employ a myriad of specialized structures for this purpose. The hair-like fimbriae or pili we encountered earlier, which act as grappling hooks, intricate adhesins, surface proteins that bind with lock and key precision to specific receptors on our cells, or even the sticky protective matrices of biofilms, which not only provide anchorage but also shield the embedded community from immune attacks and antimicrobial agents. Once adhered, some pathogens possess the remarkable ability to invade, to breach the protective barriers of our bodies and enter deeper tissues, or even to manipulate our own cells to engulf them, gaining entry into an intracellular sanctuary. This allows them to evade immune surveillance, tap into the host's resources, and spread more widely. But perhaps the most dramatic and devastating actions of many pathogens come from their production of toxins. These potent biochemical weapons are often the direct cause of the symptoms we experience during an infection. There are two primary categories: exotoxins. These are proteins secreted by living bacteria into their surroundings, often highly specific in their action and incredibly potent. Picture a molecular assassin designed to target a particular cellular process. Some neurotoxins, for example, interfere with nerve signal transmission, leading to paralysis. Others are enterotoxins, targeting the cells of the gut, causing the severe fluid loss associated with diarrheal diseases. Still others are cytotoxins, directly destroying host cells. These secreted poisons can travel far from the site of infection, causing systemic damage throughout the body. Endotoxins. In contrast, endotoxins are not secreted. Rather, they are structural components of the outer membrane of gram-negative bacteria, specifically the lipid A portion of lipopolysaccharide, LPS. They are released only when the bacterial cell dies and lyses. When released in significant amounts, endotoxins can trigger a massive, often uncontrolled, inflammatory response in the host, leading to fever, widespread inflammation, blood pressure drops, and in severe cases, septic shock, a life-threatening condition. The body's own zealous defense, triggered by these potent fragments, can inadvertently become a source of harm. Beyond adhesion, invasion, and toxin production, pathogens have evolved sophisticated strategies to evade the host immune system. Some cloak themselves in capsules, slippery outer layers that make them difficult for phagocytic immune cells to engulf. Others rapidly alter their surface proteins, a process known as antigenic variation, presenting a constantly changing face to the immune system, much like a master of disguise. Still others can actively suppress immune responses or even replicate within immune cells themselves, turning the body's defenders into unwitting hosts. This intricate dance of microbial offense and host defense underscores a fundamental truth. Our lives are in constant dialogue with the microbial world, and when this dialogue takes a turn towards conflict, our bodies are not defenseless. We are equipped with an extraordinary multi-layered system of protection, the immune system. Imagine your body as a meticulously guarded fortress, with sentinels posted at every gate and specialized brigades ready to respond to any incursion. Our immune system operates on two fundamental levels: innate immunity. This is our body's immediate, non-specific defense system, always ready for action. Think of it as the first line of defense, a rapid, generalized response to anything perceived as non-self. Physical barriers are paramount, the impenetrable fortress wall of our skin, a dry, acidic environment that few microbes can breach, the sticky mucous membranes, lining our respiratory and digestive tracts, trapping invaders, the flushing action of tears and saliva, and the acidic environment of the stomach. Should these barriers be breached, internal sentinels spring into action. Phagocytes, such as neutrophils and macrophages, are the cellular cleanup crew, engulfing and destroying invaders. Inflammation, characterized by redness, swelling, heat, and pain, is a localized response that brings immune cells and healing factors to the site of infection. Fever, a systemic increase in body temperature, can inhibit microbial growth and enhance immune cell activity. These innate responses are vital, providing immediate protection and often containing an infection before it can take hold. Adaptive immunity. This is our body's more sophisticated, highly specific and memory-based defense system. It learns and adapts. When innate immunity isn't enough, adaptive immunity steps in, recognizing specific molecular patterns, antigens, on pathogens. This system involves specialized white blood cells called lymphocytes, the B cells and T cells. B cells mature into plasma cells that produce highly specific antibodies, Y-shaped proteins that can neutralize toxins, block adhesion, and mark pathogens for destruction by other immune cells. T cells come in various forms. Some directly kill infected cells, cytotoxic T cells, while others help coordinate the entire immune response, helper T cells. The remarkable feature of adaptive immunity is its memory. Once exposed to a pathogen, the system remembers it, mounting a much faster and stronger response upon subsequent encounters. This is the principle behind vaccination. The innate and adaptive immune systems do not work in isolation. They are intricately interwoven, communicating and cooperating to provide comprehensive protection. Together, they represent a testament to billions of years of co-evolutionary struggle and adaptation between hosts and their microbial inhabitants. Yet, despite our body's formidable defenses, pathogens sometimes gain the upper hand. In such moments, humanity has sought ways to intervene, to tip the balance back in our favor. This brings us to the realm of antimicrobial strategies. The most celebrated of these interventions, and truly one of the greatest medical breakthroughs in history, is the discovery and development of their antibiotics. Born from the serendipitous observation by Alexander Fleming of mold inhibiting bacterial growth, antibiotics are compounds that specifically target and kill bacteria or inhibit their growth with minimal harm to the host. Their mechanisms of action are varied and elegant. Some, like penicillin, disrupt the synthesis of the bacterial cell wall, causing the cell to burst. Others interfere with bacterial protein synthesis, crippling their ability to build essential components. Still others target DNA replication or metabolic pathways unique to bacteria. The advent of antibiotics ushered in an era where once lethal bacterial infections became treatable, transforming medicine and public health. However, the very success of antibiotics has cast a long shadow: antibiotic resistance. Microbes, masters of adaptation and evolution, quickly developed mechanisms to circumvent these drugs. They can mutate their target sites, acquire genes that produce enzymes to break down antibiotics, or develop efflux pumps to actively expel the drugs from their cells. The widespread and sometimes indiscriminate use of antibiotics has inadvertently driven the selection for resistant strains, creating a global health crisis where common infections are once again becoming difficult or even impossible to treat. This is a stark reminder of the evolutionary power of microbes and the delicate ecological balance we perturb with our interventions. Beyond bacteria, other microbial pathogens also necessitate specific strategies. Antivirals target viruses, but their development is particularly challenging because viruses replicate within our own cells, making it difficult to harm the virus without harming the host cell. Antivirals often work by interfering with specific stages of the viral life cycle, such as attachment, entry, replication of genetic material, or assembly of new viral particles. Similarly, antifungals combat fungal infections, often by targeting components of the fungal cell wall or membrane that are absent in human cells. But perhaps the most profound and proactive antimicrobial strategy lies in prevention, the art of preparing the immune system before an encounter with a pathogen. This is the essence of vaccination. Vaccines introduce a weakened or inactivated form of a pathogen, or even just key parts of it, antigens, to the immune system. This exposure allows the adaptive immune system to leaven the pathogen's identity, to develop specific antibodies and memory cells without having to endure the actual disease. Then, should the real pathogen ever invade, the immune system is primed for a rapid, overwhelming response, often neutralizing the threat before any symptoms arise. Vaccines have eradicated diseases like smallpox and brought others like polio and measles to the brink of elimination, standing as monumental achievements in our ongoing dialogue with the microbial world. So, as we reflect on this chapter, we return to the initial question of balance. Disease in many ways is a manifestation of this balance being disturbed, whether by a highly virulent pathogen, a shift in the delicate ecosystem of our microbiome, or a momentary lapse in our immune defenses. It is a powerful reminder that we are not isolated entities, but rather complex multi-species ecosystems, constantly interacting with the vast unseen world. The struggle against pathogenic microbes is not a battle to sterilize our existence, but rather an ongoing endeavor to understand, to respect, and to restore the intricate equilibrium that defines life itself. This understanding of microbial pathogenesis and our body's defense mechanisms is not merely academic. It is foundational to medicine, public health, and our very survival. It underscores the profound interconnectedness of all life and the continuous humbling dance between host and microbe. As we move forward, we carry with us the knowledge that even in conflict, there is a deep, ancient wisdom to be found in the microbial world, guiding us towards a more holistic appreciation of health and existence. Having navigated the intricate dance of pathogens and the formidable defences of our immune system, we now stand at a new threshold, ready to explore another dimension of our relationship with the microbial world. We have seen how microbes can challenge life, but it is equally true that they can sustain and enhance it in myriad ways, often serving as our silent, tireless partners. In the Dreamy Academy, we invite you to shift your gaze from the microbial battleground to the microbial workshop, a place of extraordinary ingenuity where life's smallest engineers craft wonders for us, and often with us. This is the realm of microbial biotechnology, a testament to humanity's growing understanding and appreciation for these unseen architects. It is here that we harness their ancient wisdom and unparalleled metabolic versatility for our benefit, transforming raw materials into invaluable products, cleaning our environment, and even shaping the very fabric of life itself. Imagine, if you will, the gentle hum of fermentation, a process as old as civilization itself, yet entirely orchestrated by microscopic hands. For millennia, humanity has unknowingly collaborated with microbes to create some of our most beloved staples. Take bread, for instance. The humble yeast, Saccharomyces cerevisiae, a single-celled fungus, works its magic within the dough. As it consumes sugars, it releases carbon dioxide gas, causing the dough to rise, and alcohol, which evaporates during baking, leaving behind that distinctive, comforting aroma and texture. Without these tiny leavening agents, our bread would remain flat, dense, and lifeless. Consider the rich tapestry of dairy products that grace our tables, the tangy sharpness of yogurt, the creamy indulgence of cheese, the refreshing zest of kefir. These transformations are the masterful work of lactic acid bacteria. Species like Lactobacillus, Streptococcus, and Bifidobacterium ferment the lactose sugars in milk into lactic acid. This acid not only preserves the milk by lowering its pH, but also coagulates milk proteins, creating the distinctive textures and flavors we cherish. Each variety of cheese, each unique yogurt culture, tells a story of specific microbial communities at work, patiently crafting flavor profiles and textures that delight our senses. Even the effervescent joy of beer and the nuanced complexity of wine owe their very existence to the metabolic prowess of yeasts, converting sugars into ethanol and a spectrum of aromatic compounds that define their character. These are not merely ancient traditions, they are profound, ongoing partnerships, reminding us that collaboration with the microscopic world is woven into the very fabric of human culture. But our partnership extends far beyond the kitchen and the cellar. In the vast landscape of modern industry and medicine, microbes have become indispensable allies, their tiny factories producing a breathtaking array of substances that underpin our health and technological progress. Think back to Chapter 5 where we discussed antibiotics. It is a profound irony and a testament to microbial complexity that many of the very compounds we use to fight pathogenic bacteria are themselves produced by other microbes, primarily fungi and soil bacteria like Streptomyces. Penicillin, streptomycin, tetracycline. These life-saving molecules are the sophisticated chemical weapons of one microbe against another, which we have learned to harvest and deploy. Beyond antibiotics, microbes are now routinely engineered to produce critical pharmaceuticals. Consider insulin, a vital hormone for individuals with diabetes. Before the advent of microbial biotechnology, insulin was extracted from animal pancreases, a laborious and sometimes allergenic process. Today, thanks to recombinant DNA technology, we can insert the human insulin gene into common bacteria like Escherichia coli. These modified bacteria then become miniature insulin factories, churning out pure human-compatible insulin in vast quantities, revolutionizing treatment for millions. The same principle applies to the production of human growth hormone, interferons, used in antiviral and anti-cancer therapies, and many other therapeutic proteins. Even some vaccines, which we explored as a cornerstone of preventive medicine, are produced using microbial systems, where bacteria or yeast can be engineered to express specific antigens, triggering an immune response without causing disease. The enzymatic capabilities of microbes are another treasure trove we have begun to unlock. Enzymes are biological catalysts speeding up specific chemical reactions with incredible efficiency and specificity. Microbial enzymes are now foundational to countless industrial processes. In our laundry detergents, enzymes derived from bacteria break down stubborn stains like proteins, fats, and starches, allowing us to clean clothes more effectively at lower temperatures. In the food industry, enzymes are used to clarify fruit juices, tenderize meat, produce high-fructose corn syrup, and enhance flavors. In textiles, they soften fabrics and remove impurities. These tiny biological machines perform tasks that would be difficult, expensive, or environmentally harmful using traditional chemical methods, showcasing the elegant sustainability inherent in microbial processes. Beyond medicine and manufacturing, microbes are emerging as crucial players in our quest for a more sustainable future. The urgent need for renewable energy sources has turned our attention to the microbial world. Biofuels such as bioethanol and biogas are produced through microbial fermentation. Yeasts and bacteria convert plant biomass, agricultural waste, or even municipal waste into ethanol, which can be used as a fuel additive. Anaerobic bacteria, in the absence of oxygen, produce methane-rich biogas from organic matter, offering a clean-burning fuel source and a way to manage waste simultaneously. The potential here is immense, offering a pathway to reduce our reliance on fossil fuels and mitigate climate change. And what about the growing global challenge of plastic pollution? Here too, microbes offer a glimmer of hope. Scientists are discovering and even engineering bacteria that can degrade certain types of plastics, breaking down these persistent materials into simpler, less harmful compounds. Some microbes can even produce biodegradable bioplastics themselves, offering a sustainable alternative to conventional petroleum-based plastics. It is a slow and complex process, but the very mechanisms that allow microbes to recycle organic matter in nature are being adapted to address the synthetic materials we have introduced into our ecosystems. Perhaps one of the most compelling applications of microbial power lies in environmental stewardship, bioremediation. We have seen how microbes drive Earth's fundamental biogeochemical cycles, silently recycling nutrients and purifying our planet. Now, We are learning to leverage these natural abilities to clean up human-made pollution. When oil spills devastate marine environments, indigenous oil-eating bacteria, often stimulated by the addition of specific nutrients, proliferate and break down the complex hydrocarbons into harmless compounds like carbon dioxide and water. Similarly, microbes can be used to neutralize toxic heavy metals, degrade pesticides, and even break down radioactive waste, transforming hazardous substances into less toxic forms. It is a powerful reminder of nature's innate capacity for self-healing, facilitated by its most ancient inhabitants. The frontier of microbial biotechnology is constantly expanding, pushing the boundaries of what we once thought possible. This brings us to the exciting and sometimes challenging fields of genetic engineering and synthetic biology. We are no longer just harvesting what microbes naturally produce. We are beginning to design them. With tools like CRISPR-Cas9, a revolutionary gene editing technology originally discovered as a bacterial defense mechanism against viruses, we can precisely edit microbial genomes. This allows us to fine-tune their metabolic pathways, enhance their production of desired compounds, or even bestow entirely new functions upon them. Imagine bacteria engineered to detect specific disease markers in the human body, or yeast designed to produce flavors and fragrances that are currently difficult or costly to synthesize chemically. Synthetic biology takes this a step further, aiming to design and build entirely new biological parts, devices and systems, or even redesign existing natural biological systems. It's like programming a computer, but with DNA and proteins. Scientists are working on creating microbial biosensors that can detect pollutants with exquisite sensitivity, or microbial factories that can synthesize complex medicines from simple sugars. This field holds immense promise for addressing global challenges in health, energy, and the environment. As we stand at this fascinating precipice of microbial design, we are also reminded of the profound ethical and societal considerations that accompany such power. With the ability to reshape life at its most fundamental level comes the responsibility to do so wisely, thoughtfully, and with a deep understanding of potential consequences. We must consider biosafety, the equitable distribution of benefits, and the long-term impact of introducing engineered organisms into our ecosystems. The journey through the microbial world is one of continuous revelation, from the ancient foundational simplicity of prokaryotes to the intricate dance of microbial communities, from their relentless adaptability in extreme environments to their dual roles as agents of disease and indispensable partners in health and industry. Microbes constantly challenge our perceptions and expand our understanding of life itself. They are not merely tiny entities. They are the bedrock, the engine, and increasingly the blueprint for a future we are only just beginning to imagine. As we conclude this exploration of microbial fundamentals, we hope you carry with you not just knowledge, but a renewed sense of wonder. The invisible universe teems with life, intelligence, and purpose. It whispers stories of billions of years of evolution, of interconnectedness, and of an enduring resilience that continues to shape our world. The Dreamy Academy invites you to keep your curiosity alive, to continue observing, questioning, and marveling at the unseen architects who orchestrate the grand symphony of life, forever influencing our existence in ways both profound and subtle. The adventure of discovery, we assure you, is never truly over. As we journey deeper into the intricate dance between humanity and the microbial world, we arrive at a frontier shimmering with both boundless promise and profound responsibility. It's a place where the air hums with the silent energy of discovery, a space where the known meets the imagined, and the very fabric of life seems to whisper secrets into the ears of those bold enough to listen. We have seen how these unseen architects have shaped our planet, sustained our lives, and even offered solutions to some of our most pressing challenges. From the ancient art of fermentation, which transformed simple grains into nourishing breads and beverages, to the modern marvels of bioremediation, where microbes consume pollutants, and the intricate production of life-saving pharmaceuticals. But what happens when we move beyond merely harnessing their existing capabilities, beyond observing their natural genius, and begin to design them? What happens when we become not just passive observers, but active co-creators, hands-on sculptors in the microbial realm? The question hangs in the air, weighty and exhilarating, a testament to our insatiable curiosity and ever-growing technological prowess. This, dear explorer, is the realm of genetic engineering and synthetic biology, where the very blueprint of life becomes a canvas for our ingenuity, and the microbes, our most ancient and adaptable companions, transform into programmable partners in a future we are only just beginning to envision. It's a journey into the microscopic, yes, but one with macroscopic implications, challenging our perceptions of nature, technology, and our place within the grand tapestry of existence. Imagine, if you will, the elegant simplicity of a prokaryotic cell, its genetic code a finely tuned instruction manual, written in the universal language of DNA. For millennia, this manual has been edited by the slow, deliberate hand of evolution, a process of trial and error spanning eons, refining life with painstaking patience. Now, with the sophisticated tools of genetic engineering, we can read, understand, and even rewrite specific chapters of that manual with astonishing speed and precision. We can introduce entirely new instructions, delete redundant or harmful ones, or rearrange entire paragraphs of genetic information, all with a control that would have been deemed pure science fiction just a generation ago. It's like being handed the master code to a complex self-replicating machine and finding you have the power not just to repair it, but to upgrade its very operating system. This isn't just about tweaking existing functions, it's about imbuing microbes with entirely new capabilities, turning them into bespoke biological machines tailored to specific human-defined tasks. A scientist hunched over a microscope might feel a quiet thrill of power, a sense of profound responsibility, as they meticulously pipette a solution containing a newly engineered plasmid, knowing that within that tiny drop lies the potential for a revolution. Just one more base pair, she might whisper to herself, her brow furrowed in concentration, and the pathway will be complete. Synthetic biology takes this a significant step further. If genetic engineering is akin to editing a novel, carefully correcting grammar, refining plot points, or adding new subplots, then synthetic biology is like writing an entirely new story, or even designing a new language from scratch. It's about applying rigorous engineering principles, design, abstraction, standardization to the messy, complex world of biology. This involves designing and building novel biological parts, devices and systems from the ground up, and even redesigning existing natural biological systems for entirely new and useful purposes. We are learning to speak the language of life with a fluency that would have been unimaginable just a few decades ago, moving from simple, incremental modification to intentional, rational design. It requires a different kind of mindset, one that marries the curiosity of a biologist with the systematic approach of an engineer constantly asking, can we build this? And if so, how can we build it better, more predictably, more reliably? Consider the revolutionary power of CRISPR, a name that echoes through the halls of modern biology like a whispered secret of profound potential, a discovery that has fundamentally reshaped our capabilities. CRISPR, or clustered regularly interspaced short palindromic repeats, is not an artificial construct but a naturally occurring bacterial immune system, a marvel of biological defense. It acts like a molecular scissor, guided by a precise RNA molecule that microbes use to identify and snip out invading viral DNA, protecting themselves from infection. Scientists have ingeniously repurposed this ancient defense mechanism, transforming it into an incredibly precise and versatile gene editing tool. With CRISPR, we can now target specific sequences within a microbe's genome with unprecedented accuracy, cutting, pasting, and inserting genetic material with exquisite control, literally rewriting the code of life with a precision that feels almost surgical. The moment of discovery, the realization of its potential, must have been an electrifying jolt for the researchers involved, a paradigm shift unfolding in real time. What does this mean for our microbial partners, for the myriad bacteria and fungi that share our world? It means we can program bacteria to produce novel biofuels more efficiently, transforming agricultural waste or even atmospheric carbon into clean energy with remarkable speed and minimal environmental footprint. Imagine a future where fuel is literally grown, not drilled, where the air smells cleaner and the sky is clearer, all thanks to microscopic factories. We can engineer them to detect specific disease markers in our bodies with far greater sensitivity and earlier detection capabilities than many current diagnostic tools, essentially turning them into microscopic living biosensors that patrol our internal landscapes. A young intern might exclaim, holding up a tablet displaying complex data. Imagine a future where a benign, engineered bacterium, ingested as part of a daily probiotic, could detect the earliest signs of cancer or inflammation in your gut, and then, with pinpoint accuracy, release a therapeutic compound directly at the precise site of concern, long before symptoms even manifest. The possibilities extend far beyond medicine and energy. We are designing microbes to produce sustainable materials, perhaps even growing 16 that fully degrade in nature, offering a tangible solution to one of our planet's most pervasive environmental crises. Picture a world where packaging dissolves harmlessly back into the earth, leaving no trace. We can engineer them to capture carbon dioxide directly from the atmosphere, turning a harmful greenhouse gas into a valuable resource for industry or agriculture, effectively reversing the tide of climate change. Or consider their potential to break down persistent pollutants in contaminated soils and waters with unparalleled efficiency, transforming toxic industrial sites into fertile ground once more. These tiny architects, once solely guided by the blind hand of natural selection, are now becoming the conscious designers of a cleaner, more sustainable future, their metabolic pathways rerouted and optimized for the grand challenges of our era. The vision is breathtaking, almost utopian in its promise. Yet, with such profound power comes an equally profound responsibility, as we learn to sculpt the very fabric of life. To imbue it with new instructions and purposes, we must pause, reflect, and ask ourselves, what are the ethical boundaries of this endeavour? What are the potential, unintended consequences of unleashing these meticulously engineered organisms into complex, interconnected ecosystems that we still only partially understand? These are not questions that can be answered lightly or in isolation. One of the foremost concerns, a constant hum of caution in the minds of researchers, is the safety and containment of these meticulously designed microbes. While many applications involve contained environments, bioreactors, laboratories, specific industrial settings, the potential for unintended release, however small or improbable, demands rigorous protocols, multi-layered safety measures, and constant vigilance. What if a bacterium engineered for bioremediation, specifically designed to thrive only in a highly polluted site, were to escape its intended containment? Could it outcompete beneficial native species in an uncontaminated environment, disrupting a delicate ecological balance? The intricate balance of microbial communities, honed over billions of years, is incredibly resilient but also delicately poised. We have triple redundancy on the filtration system, Dr. Chen, a lab technician might reassure her supervisor, and the genetic kill switch is programmed for nutrient deprivation. Dr. Chen nods, but a flicker of worry crosses her face. It's not enough to be careful, she thinks. We must be infallible. We must approach such interventions with humility and a deep understanding of ecological dynamics, acknowledging the vast unknowns. Then there is the question of environmental impact. Even if perfectly contained, the products generated by engineered microbes might have unforeseen effects on surrounding ecosystems or human health. And what if we succeed in creating highly efficient novel metabolic pathways that process nutrients or produce compounds at rates far exceeding natural processes? Could these disrupt established biogeochemical cycles in ways we cannot yet fully predict, altering the fundamental chemistry of our planet? The interconnectedness we have explored throughout our journey reminds us that every action, however small, ripples through the vast web of life, often in ways we can't fully grasp until it's too late. The biosecurity and dual-use dilemma also looms large, casting a long shadow over the brightest promises. The very same technologies that allow us to engineer microbes for immensely beneficial purposes, to produce life-saving medicines, to clean pollution, to create sustainable food sources, could in theory be twisted for malicious intent. They could be used to create novel pathogens with enhanced virulence or resistance, or to weaponize existing ones. This necessitates robust international regulations, transparent research practices, and a global commitment to ethical scientific conduct that transcends national borders and competitive pressures. Scientists working in this field carry this heavy burden, a constant awareness of the double-edged sword they wield. Knowledge is power, a researcher might muse during a sleepless night. But it's also a terrifying responsibility. how do we ensure it's always used for good? And as we ponder the creation of entirely new biological systems, even the theoretical possibility of synthesizing life from fundamental chemical building blocks, we touch upon profound philosophical questions. What does it truly mean to create life? To design an organism from the ground up that can replicate and evolve? What are our moral and ethical responsibilities to these designed organisms, especially if they gain unforeseen complexities? Who owns the intellectual property of a living, breathing, self-replicating entity engineered in a lab? Are we playing God, or merely understanding and manipulating the rules of a cosmic game? These are not questions with easy answers, and they demand careful, inclusive societal discourse, involving not just scientists and ethicists, but philosophers, policy makers, and the public at large. Our journey through microbiology has consistently revealed the humility required to truly understand life. We are but a small part of a vast, ancient, and incredibly complex biological tapestry. As we gain the power to weave new threads into this tapestry, to introduce novel patterns and functions, we must do so with immense respect for what came before, foresight for what might come next, and a profound sense of stewardship for all life. The promise of genetically engineered and synthetically designed microbes is immense, offering paths to unprecedented sustainability, improved health, and a deeper understanding of life itself. But this promise is inextricably linked to our collective wisdom, our ethical compass, and our unwavering commitment to responsible innovation. The Dreamy Academy invites you not just to marvel at the wonders of the microbial world, but to engage actively with its future, to contemplate its profound implications, and to participate in the ongoing conversation about how we, as a species, will wield the extraordinary power we are beginning to unlock. The unseen architects continue their work, and now we have the opportunity to stand beside them, not as masters seeking to dominate, but as thoughtful partners, shaping a future where life, in all its myriad and magnificent forms, can flourish. The exploration never truly ends. It merely evolves, inviting us to ever deeper layers of understanding and wonder, urging us forward with a blend of scientific rigor and profound reverence. Even as we wield the exquisite tools of genetic engineering, shaping microbial destinies with precision and purpose, we are reminded of the profound depths that remain unexplored, the silent majority of life that continues to whisper its secrets just beyond our grasp. Our journey through the microbial realm has illuminated its ancient origins, its fundamental architectures, its pervasive influence, and its vast potential for both healing and harm. Yet, for all our advancements, for all our burgeoning understanding, we stand at the precipice of an even grander unknown. Imagine, if you will, that for every star we can see in the night sky, there are 1,000,000 more, veiled in the cosmic dust, their light yet to reach our eyes. So it is with the microbial world. For centuries, our understanding was limited to those organisms we could coax into growth within the sterile confines of a petri dish. We nurtured them, observed their colonies, and marveled at their visible transformations. But this approach, while foundational, revealed only a minuscule fraction of the true microbial diversity. This unseen majority, this vast uncultured kingdom, has come to be known as the microbial dark matter. It is a testament to the elegant simplicity and profound strength of prokaryotic life that so many species have resisted our attempts at domestication, thriving in their intricate natural niches, stubbornly refusing to conform to the conditions of our laboratories. We speak of the incredible efficiency, rapid reproduction, and adaptability of prokaryotes, and nowhere is this more evident than in their ability to exist in forms and functions we are only just beginning to comprehend. They are the fundamental blueprint, the original design, and their persistence in environments we deem hostile or inaccessible is a constant source of wonder. To peer into this dark matter, we have had to invent new eyes, new ways of listening to the whispers of life. This is where the quiet revolution of metagenomics begins. Instead of trying to grow individual microbes, we now extract all the DNA directly from an environmental sample, be it a handful of soil, a litre of ocean water, a swab from your skin, or a core from ancient ice. We then sequence this vast library of genetic information, like reading countless individual stories from an ancient interconnected manuscript. It's a profound shift in perspective, allowing us to reconstruct the genetic potential of entire microbial communities, even if we have never seen a single one of their cells divide in a lab. With metagenomics, we no longer need to isolate and culture. We are, in essence, listening to the collective hum of a bustling metropolis, discerning the different languages spoken, the various trades practiced, and the unique contributions of each inhabitant, all without ever stepping foot inside their individual homes. We are uncovering entirely new phyla of bacteria and archaea, organisms whose very existence challenges our previous classifications and expands our understanding of life's tree. We find metabolic pathways we've never encountered, enzymes with unprecedented capabilities, and ecological interactions of bewildering complexity. This approach reveals the true architects and silent engineers of our planet, working tirelessly, often unseen, in a symphony of biochemical reactions that sustain all macroscopic life. From the grand tapestry of communities, our gaze also narrows, seeking to understand the unique melody of each individual player. This is the realm of single-cell microbiology, a frontier that allows us to dissect the heterogeneity within a seemingly uniform population. Imagine a bacterial colony appearing identical to the naked eye. Yet, beneath the surface, individual cells might be expressing different genes, responding to subtle environmental cues in unique ways, or even adopting specialized roles within the collective. By isolating and analyzing the genetic material or protein expression of single cells, we unlock a deeper understanding of microbial individuality, adaptation, and the division of labor that underpins their collective strength. It's like moving from studying the behavior of an entire human crowd to understanding the motivations and actions of each person within it. This precision allows us to explore how individual microbes cope with stress, resist antibiotics, or contribute to complex diseases, revealing the nuanced strategies that allow them to thrive. As our tools become more refined, so too does our appreciation for the intricate dance between microbes and their environments, stretching beyond the immediate and into the global. The ancient, pervasive presence of these tiny architects means their influence is woven into the very fabric of our planet's future. We are increasingly recognizing the critical role of microbes in climate change, both as drivers and as potential solutions. From the methane-producing archaea in wetlands and thawing permafrost to the vast photosynthetic cyanobacteria in our oceans that sequester carbon dioxide, microbes are the unseen regulators of atmospheric gases. Understanding their responses to a warming world, how they might shift their metabolic activities, and how we might harness them to mitigate environmental damage, represents one of the grandest challenges and opportunities of our time. They are, in essence, the planet's original environmental engineers, and their ongoing work dictates the habitability of Earth. Our explorations also extend beyond Earth itself. The resilience and adaptability of Earth's microbes, their ability to thrive in extreme conditions, from scalding hydrothermal vents to freezing polar ice, from acidic hot springs to radiation-drenched environments, fuels the captivating field of astrobiology, or exomicrobiology. If life can persist in such harsh niches on our own world, what possibilities exist on distant celestial bodies? The search for microbial life on Mars, Europa, or Enceladus is not merely a quest for extraterrestrial organisms. It is a profound philosophical inquiry into the universality of life, its potential forms and its fundamental principles. Could the elegant simplicity of prokaryotic life, the original design, be replicated across the cosmos? This study of extremophiles on Earth provides invaluable insights into the biosignatures we might seek, guiding our probes and telescopes to the most promising corners of the universe, reminding us that the story of life might be far grander and more widespread than we once imagined. Closer to home, the pressing challenge of antimicrobial resistance continues to spur innovation. While we previously discussed the mechanisms of resistance and the need for new antibiotics, the frontiers of discovery are now exploring entirely novel strategies. This includes revisiting ancient remedies, like phage therapy, where viruses that specifically target and destroy bacteria are deployed as living medicines. It also involves delving into the complex chemistry of microbial communication, seeking to disarm pathogens by disrupting their ability to coordinate attacks rather than outright killing them. We are learning to think like microbes, to understand their social lives, their chemical languages and their vulnerabilities, moving beyond a simplistic kill or be killed approach to a more nuanced ecological intervention. And within our own bodies, the human microbiome remains a universe of largely unmapped territories. While we understand its general importance, the precise mechanisms by which specific microbial communities influence individual health, disease susceptibility, and even mood are still being unraveled. The future promises personalized microbiome interventions, where our unique microbial fingerprint guides, dietary recommendations, probiotic therapies, and even drug selection. We are moving towards a future where understanding your unique microbial ecosystem is as fundamental to your health as knowing your blood type. The journey through the microbial realm is an unending odyssey, a continuous cycle of discovery and humility. Every answer we uncover, every new technology we develop, inevitably births 100 new questions. We are constantly reminded of the intricate interconnectedness of all life, the profound wisdom encoded in the earliest forms, and the delicate balance that sustains our world. The more we learn, the more we realize the vastness of what we do not yet know, fostering a deep sense of reverence and an insatiable curiosity. This is the essence of microbiology, an eternal invitation to explore, to wonder, to understand the unseen architects and silent engineers that have shaped life from its very inception and continue to orchestrate its future. It is a journey that challenges our perceptions, expands our imagination, and ultimately humbles us before the elegant, enduring power of the very smallest forms of life. The whispers from the unseen world continue, inviting you to listen, to learn, and to join in this magnificent unfolding adventure. As our peaceful exploration through the unseen realms of microbiology begins to draw towards a contemplative close, we find ourselves standing at the precipice of a profound understanding. We have journeyed through billions of years, from the primordial whispers of the first prokaryotic cells to the intricate dance of modern microbial communities, from the silent architect shaping our planet to the cutting-edge laboratories where life itself is being reimagined. This adventure, you see, is not merely about accumulating facts. It is about shifting our perspective, about truly seeing the world through a microbial lens. We began by marveling at the elegant simplicity of prokaryotes, the fundamental blueprint, the original design. Their profound strength, incredible efficiency, and dazzling adaptability laid the very foundation for all that was to follow. From these ancient roots sprang the grander, more complex structures of eukaryotes, their sophisticated internal architecture enabling the specialization that led to the wondrous diversity of multicellular life. Each step of this evolutionary ballet, we now understand, was choreographed by microbes, a testament to their enduring ingenuity. You have witnessed their extraordinary resilience, thriving in conditions that defy our imagination, the scorching vents, the freezing poles, the caustic depths. These extremophiles, these tenacious pioneers, not only survive but sculpt the very geochemistry of our world, orchestrating the vital biogeochemical cycles of carbon, nitrogen, sulfur, and phosphorus. Without their tireless work, the breath we draw, the food we eat, the very ground beneath our feet would be utterly unrecognizable. They are the planet's silent engineers, maintaining the delicate balance that sustains us all. We have peered into the bustling metropolis that is your own body, the human microbiome, a vast and intricate ecosystem of trillions of microbial partners. We've seen how they digest our food, educate our immune systems, and even influence our moods. This intimate coexistence shatters the illusion of our individuality, revealing us as composite beings, inextricably interwoven with the microbial world. And yes, we've confronted the shadows, the pathogens, those formidable adversaries whose virulence factors challenge our defenses, reminding us of the constant evolutionary arms race that shapes life. Yet even here, our understanding of their mechanisms has led to powerful strategies. The antibiotics, the antivirals, the vaccines, and the ever-evolving quest to overcome resistance. Our journey has also taken us into the realm of innovation, where humanity, inspired by microbial genius, harnesses these tiny powerhouses for our own ends, from the age-old art of fermentation to the industrial production of life-saving pharmaceuticals, from sustainable biofuels to the bioremediation of polluted landscapes. Microbes are our most versatile collaborators, and then the true frontier, genetic engineering and synthetic biology. With tools like CRISPR, we now possess the unprecedented ability to precisely edit microbial genomes, to design new biological systems, to program life itself. This power, as we discussed, opens doors to unimaginable possibilities: enhanced carbon capture, living biosensors, sustainable materials, but it also casts a long shadow of ethical dilemmas, biosecurity concerns, and profound philosophical questions about our role as creators. In our last chapter, we acknowledged the vast microbial dark matter, the uncultured majority that still eludes our grasp. Metagenomics and single-cell microbiology are just beginning to illuminate this hidden diversity, revealing new pathways, new chemistries, new forms of life we never imagined. We spoke of microbes' critical role in climate change, their potential in astrobiology, and the promise of personalized microbiome interventions. The journey, we concluded, is continuous, humbling, and endless. Now, let us draw these threads together to see the grand tapestry that has emerged. What does this deep dive into the microbial world ultimately teach us about life itself, about our place within it, and about the future we are collectively weaving? First, and perhaps most profoundly, microbes reveal the fundamental unity of all life. Despite the dizzying diversity we observe from the simplest archaeon to the most complex redwood, the underlying molecular machinery is remarkably conserved. The genetic code, the elegant dance of DNA and RNA, the protein synthesis, these are universal languages spoken by every living cell. This shared heritage, forged in the microbial crucible billions of years ago, underscores our deep evolutionary kinship. We are not separate from the microbial world. We are an intricate, sophisticated extension of it. Our cells, with their mitochondrial powerhouses, carry the echoes of ancient endosymbiosis, a microbial partnership that made complex life possible. You are quite literally a walking, breathing testament to microbial collaboration. This understanding fosters a deep sense of humility and interconnectedness. We, with our grand civilizations and technological prowess, are utterly dependent on these unseen architects. Every ecosystem, every breath, every cycle of nutrient flow relies on their tireless, often invisible work. To truly grasp this is to shed the anthropocentric view and embrace a more holistic perspective, recognizing ourselves as but one thread in an unimaginably vast and ancient web of life. It invites us to consider our actions not just in terms of their immediate human impact, but in their ripples across the microbial ecosystems that underpin everything. The microbial world also teaches us about resilience and adaptability beyond measure. Life, in its microbial forms, has faced every conceivable challenge over billions of years: catastrophic extinctions, dramatic climate shifts, cosmic radiation. And yet, it persists, it evolves, it finds new ways to thrive. This enduring spirit, this relentless drive to find a niche and flourish, is an inspiring lesson in the sheer tenacity of existence. It suggests that even in the face of our own global challenges, there is an inherent capacity for life to endure, provided we act with wisdom and respect for its fundamental principles. As we stand on the cusp of truly designing and engineering life, the lessons of microbiology become even more critical, informing our higher responsibility as stewards and co-creators. We now possess the power not just to observe, but to intervene, to direct evolution, to build new biological futures. This power demands profound ethical reflection. What are the long-term consequences of releasing engineered microbes into the environment? How do we ensure equity in access to microbial biotechnologies? How do we safeguard against unintended consequences or malicious misuse? The very complexity and interconnectedness of microbial systems, which we have painstakingly explored, underscore the need for caution, foresight, and a deep respect for the intricate balance of nature. We are not merely manipulating isolated entities, we are subtly altering the grand tapestry of life itself. And what of the universe beyond our Earth? The principles we have learned from our planet's microbes, their incredible adaptability, their metabolic versatility, their ability to thrive in extreme conditions, provide a powerful framework for astrobiology. If life exists elsewhere, it is highly probable that it too will manifest in microbial forms first. Perhaps the elegant simplicity of prokaryotes, their efficient energy capture and rapid reproduction, represents a universal blueprint for nascent life across the cosmos. When we gaze at the stars, searching for signs of other worlds, we are now armed with a microbial lens, understanding the fundamental conditions and chemical signatures that might betray the presence of unseen architects on distant planets. Our humble microbes, in essence, expand our imagination for what life can be, where it can exist, and how it might manifest in the vastness of space. So, as this chapter unfolds, it is not an ending, but rather an invitation to a deeper, ongoing engagement with the microbial world. The Dreamy Academy has offered you a glimpse, a foundation, a spark. But the true adventure lies in carrying this microbial lens into your everyday life, into your future contemplations, into your professional endeavours, to see the world anew, to recognize the unseen architects in the soil beneath your feet, in the food on your plate, in the very air you breathe, and within yourself, is to unlock a profound appreciation for the intricate dance of existence. The journey through microbiology is a journey into the heart of life itself. It is a humbling reminder that the most powerful forces, the most ancient wisdom, often reside in the smallest, most unassuming forms. It is a call to wonder, to question, and to continue exploring the infinite mysteries that still lie hidden, waiting to be discovered. For even with all we have learned, the microbial dark matter persists, beckoning us onward, whispering of new chemistries, new ecologies, new possibilities yet unimagined. May this understanding foster within you a renewed sense of connection, a profound respect for the delicate balance of our planet, and an enduring curiosity for the unseen wonders that continue to shape our world, moment by moment, breath by breath. The microbes, the true masters of this planet, continue their work, and so too must our journey of discovery. Our journey through the microscopic realms has unfolded like an ancient scroll, revealing wonders at every turn. We have traversed the elegant simplicity of prokaryotes, marvelled at their profound strength and adaptability, and witnessed their intricate dance with the grander structures of eukaryotes. We've seen them sculpt our world, sustain our bodies, and even inspire our greatest technological advancements. Yet, as with any truly vast and ancient landscape, for every peak we have scaled, countless valleys remain shrouded in mist, hinting at mysteries yet to be unveiled. This, dear explorer, is where our current chapter begins, peering into the luminous fog of the unknown, acknowledging the boundless frontiers that still beckon us in the world of microbiology. Our understanding, though ever growing, remains but a flickering candle in the immense darkness of what we do not yet comprehend. Consider the phenomenon we affectionately call microbial dark matter. Imagine, if you will, that for centuries we have only been able to study the creatures willing to wander into our brightly lit human cities. The vast majority of life, the teeming populations of the deep wilderness, the denizens of the most remote mountains and ocean trenches, have remained elusive, unseen. This is precisely the challenge we face with microbes. For a long time, our primary method of studying them involved culturing them in laboratories, essentially inviting them to grow in our specific, often limited conditions. What we discovered, startlingly, is that over 99% of microbial species refuse our invitation. They are the uncultured majority, the silent, invisible inhabitants of nearly every environment on Earth, whose lives and secrets have largely remained beyond our grasp. But the light of human ingenuity, coupled with an insatiable curiosity, is beginning to pierce this veil. We are no longer solely reliant on coaxing microbes to grow in petri dishes. Instead, we are learning to listen in on their conversations, to decode their very essence without ever needing to see them directly. This revolution is powered by groundbreaking techniques like metagenomics and single-cell microbiology. Metagenomics is akin to sifting through the genetic library of an entire ecosystem. Instead of isolating individual microbes, we extract all the genetic material, the DNA, directly from an environmental sample, be it a scoop of soil, a drop of ocean water, or a swab from your own skin. We then sequence this vast collection of genetic fragments, piecing together the blueprints of countless organisms, many of which we've never encountered before. It's like finding fragments of thousands of books in an ancient ruin and, through careful analysis, reconstructing the stories they tell, even if the authors themselves remain hidden. This approach has unveiled an astonishing diversity, revealing entire phyla of bacteria and archaea that exist only as genetic signatures, hinting at metabolic pathways and ecological roles we are only just beginning to imagine. Building upon this, single-cell microbiology allows us to zoom in even further, isolating and analyzing the genetic material from individual microbial cells one by one. This is like picking out a single unique book from that vast ruined library and reading it cover to cover. It provides unprecedented detail, revealing the specific functions and adaptations of individual organisms within a complex community, offering clues about how they survive, interact, and thrive in their native habitats. Together, these tools are transforming our understanding of life on Earth, revealing a hidden biological richness that dwarfs anything we previously imagined. We are discovering new enzymes with industrial potential, novel antibiotic compounds, and entirely new branches on the tree of life, prompting us to redraw our maps of existence. These revelations about microbial dark matter have profound implications, particularly as we confront the most pressing global challenges of our era. Consider climate change, a phenomenon driven by the intricate dance of gases and energy within Earth systems. Microbes, those ancient engineers, are central to this dance. They are the primary drivers of the carbon cycle, transforming atmospheric CO2 into organic matter and then breaking it down again, releasing methane, a potent greenhouse gas, or sequestering carbon in soils and oceans. As our planet warms, as ocean chemistry shifts, and as ecosystems transform, the microbial communities within them will respond, and their responses will, in turn, feed back into the global climate system. Understanding this complex interplay, identifying which microbes are producing more methane in thawing permafrost, or which are more efficient at carbon sequestration in marine environments, is not merely an academic exercise. It is critical for predicting future climate scenarios and for developing microbial-based strategies to mitigate climate change, perhaps through engineering more efficient carbon-capturing microbes or suppressing methane-producing ones. And what of life beyond Earth? The profound resilience of extremophiles, their capacity to thrive in conditions we once deemed utterly hostile, has fundamentally reshaped our search for astrobiology. If life can flourish in boiling acid, beneath miles of ice, or in the vacuum of space, then the possibilities for life on other planets or moons expand exponentially. Our understanding of Earth's extremophiles provides a crucial roadmap, guiding our search for biosignatures on Mars, Europa, or Enceladus, places where liquid water might exist, and where microbial life, if it exists, would likely resemble the hardy, adaptable forms we find in Earth's most challenging environments. What a humbling thought, that the very organisms that laid the foundation for life on our home planet might also be the most likely forms of life we encounter in the vast cosmos. Closer to home, the ongoing struggle against antimicrobial resistance remains a formidable challenge. The very adaptability that makes microbes so awe-inspiring also makes them formidable adversaries. As we continue to face the diminishing efficacy of traditional antibiotics, the need for innovative solutions becomes ever more urgent. One promising avenue, a return to nature's own arsenal, is phage therapy. Bacteriophages, or phages, are viruses that specifically target and infect bacteria. They are the most abundant biological entities on Earth, constantly engaged in a microscopic arms race with their bacterial hosts. By carefully selecting and deploying specific phages, we can develop highly targeted treatments that destroy pathogenic bacteria while leaving beneficial microbes unharmed. A precision strike compared to the broad-spectrum approach of many antibiotics. This ancient biological warfare, refined over billions of years, offers a compelling new weapon in our fight against resistant infections. Similarly, our understanding of the human microbiome is advancing at an astonishing pace, moving us towards a future of personalized microbiome interventions. We are learning that each of us carries a unique microbial fingerprint, an internal ecosystem as individual as our actual fingerprints. This intricate community of bacteria, fungi, and archaea profoundly influences our digestion, immunity, metabolism, and even our mental health. Imagine a future where, instead of generic health advice, interventions are tailored precisely to your unique microbial profile. Dietary recommendations could be optimized to nourish your specific gut flora. Probiotic therapies could be designed to introduce specific beneficial strains. Even fecal microbiota transplants, already showing promise for treating recurrent C. Difficile infections could become more precisely engineered to restore balance to a dysbiotic system, offering hope for conditions ranging from inflammatory bowel disease to obesity and neurological disorders. Yet, as our capabilities grow, particularly in the realm of ungenetic engineering and synthetic biology, so too do our responsibilities. The power to reprogram microbes, to design and create new forms of life with specific functions, is a double-edged sword that demands profound ethical consideration. We stand at a precipice where we can not only observe and understand life, but actively reshape it. The ethical dilemmas are multifaceted and complex. First, there are concerns about biosafety and containment. What are the risks of releasing engineered microbes into the environment? Could they outcompete natural strains, transfer their engineered genes to other organisms, or cause unintended ecological disruption? Rigorous containment protocols and cautious environmental risk assessments are paramount to prevent unforeseen consequences. The potential for environmental impact is not to be underestimated. Ecosystems are delicately balanced, and even seemingly minor alterations could ripple through the web of life. Then there are the biosecurity concerns, often referred to as the dual-use dilemma. The very technologies that can create beneficial medicines or sustainable fuels could, in the wrong hands, be weaponized. The ease with which genetic editing tools like CRISPR can be acquired and utilized raises questions about regulating access and preventing malicious use. This necessitates robust international frameworks, open scientific dialogue, and a global commitment to responsible innovation. Finally, we grapple with deeply philosophical questions. What does it mean to create life? Do we have the right to own or patent newly engineered organisms? Where do we draw the line between therapeutic intervention and fundamental alteration of life? What are the long-term implications for biodiversity and the natural world if we increasingly rely on designer microbes? These are not questions with easy answers, and they require ongoing, thoughtful deliberation from scientists, ethicists, policymakers, and the public alike. Our journey through microbiology, then, is not merely a scientific endeavour. It is a continuous philosophical exploration. It demands not just intellect, but also humility. The deeper we delve into the microbial world, the more we realize the fundamental unity of all life, the intricate interconnectedness that binds us to the simplest bacterium and the most ancient archaeon. We are reminded that our existence is predicated on theirs, that the air we breathe, the food we eat, the very health of our planet, are all meticulously maintained by these unseen architects. As we stand on the cusp of truly unprecedented understanding and unprecedented power, our role evolves. We are no longer just observers nor simply beneficiaries. We are becoming, through our knowledge and our technologies, stewards and co-creators of the microbial world. This role carries with it an immense responsibility to guide this future with wisdom, with reverence for life in all its forms, and with an unwavering commitment to the well-being of our planet and all its inhabitants. The quiet hum of the microbial universe continues, a symphony billions of years in the making. Our invitation to listen, to learn, and to participate in its ongoing creation remains open. May our curiosity never wane, and may our stewardship be guided by the profound wonder that this invisible world so generously offers. The adventure, dear friend, has only just begun.